This conference will now be recorded.
Okay, so disease is caused by bacteriophages. So what is this? The bacteriophages usually infect bacteria only, but they directly do not cause any disease. However, when they infect bacteria, they are able to convert virulent bacteria into virulent strains. Some of the examples are cholera. Second one is scarlet fever. Scarlet fever is usually caused by streptococci. When streptococci is usually infected by T12 bacteriophage, then it may cause cancer. Okay. Next, morphology of bacteria. This morphology. Bacteriophages usually tadpole shape consisting of about hexagonal head and hexagonal head. Head is called as hexagon because it usually consists of both sides. And tail is round. And within the head, there is the chain of this. At the base of the tail, there is a base plate, which is usually required for penetration. And tail cover, which usually help in attaching the bacteriophage to the surface of bacterial cells. These bacteriophages are used for cloning genes among other applications. Proteins and peptides are fused to the capsid of the phage. And the combination of a phage and a peptide is known as fusion protein. Next, types of phages. There are so many types of bacteriophages. T-event phages such as T2, T4, and T6 are the ones which usually infect Escherichia coli. Another phage called as temperate phage, such as lambda and mu. Spherical phages with single-stranded DNA, such as PX174. Filamentous phages, such as M13, and RNA phages, such as Q beta. So, these are all the different types of phages. Life cycle of bacteriophage. Bacteriophages usually exhibit two types of life cycle. They are virulent or life cycle. Second one is temperate or lysogenic. Okay. So two types of life cycle are there, light and lysogenic. Light is also called as virulent cell. Lysogenic, temperate cell. Virulent cell. It is when the growth results in the lysis of the host and the result of the protein phase. So, in this cycle, there is lysis of means there is the death of the host in the cell in which the replication. That's why it is called as virulent. It is destroying the host. So, the cycle is known as virulent. How does it take place? Usually, the tail of a bacteriophage consists of a core, a tail sheath, base plate, tail fins, and tail covers, all of which are composed of different proteins. The tail and related structures of bacteriophage are generally involved in attachment of the phage and securing the entry of viral nucleic acid into the host. Before viral infection, the cell is involved in the replication of its own DNA, transcription, and finally, translation of its own genetic material involving in growth and cell division. Okay, so what is this? What is happening in life? Okay, you know the tail usually consists of sheet, base plate. On the base plate, there are fins and tail fibers. Okay, all of these structures of the tail are involved in attachment of bacteriophage to the host cell. Once it attaches to the host cell, so that the viral nucleic acid can enter into the host cell. After infection, the viral DNA usually takes things of the host cell and uses it to produce the nucleic acid and proteins needed for production of new host proteins. Viral DNA replaces the host DNA as a template for bone research. The steps which are usually involved in life cycle or build cycle are adsorption, penetration, synthesis of phage components, assembly, maturation, and release of progeny phage particles. First one, adsorption. What is adsorption? The phage particles come into contact with the bacterial cells by random collision and attaches to the surface of a susceptible bacterial virus strain. This is a specific process and depends upon the presence of receptor sites on the bacterial cell for the terminal base place of it. Cofactors like adherence are also necessary for adsorption. Okay, so the first step in life cycle is adsorption. Means what? It has to come to the surface of the bacterial cell and it has to attach to the surface of bacterial cell. This attachment is a specific process and it also depends upon the specific receptors which are present on the surface of the bacterial cell to which it wants to get attached. Next one, penetration. So what will happen after attachment? After attachment, the phage will penetrate the bacteria. How does it penetrate the bacteria? How does it attach firmly? The base plates are there, and fins are there, and fibers are there. Right? Next, what will happen? The phage DNA is injected into the bacteria to their halo core and infection of the bacterium by a nucleic acid, usually known as transfection. The base plates and tail fibers are held firmly against the bacterial cell, and the hollow core pierces the cell wall. Okay, so for example, bacteriophage. Okay, so you see if this is a bacteriophage. Okay, so this is the example. This is a bacteriophage example. Okay, so on the end of this wall is centered present, and this is bacterial cell. So what does it do? This bacteriophage is first of all, this has to attach, right? It has to attach. Once it attaches, what will happen? This is a tail, right? Inside the tail, here the core is present. This core, okay, places inside the bacterial cell. Okay, once it present inside the bacterial cell, what will happen? The genetic material of this DNA, which is usually present in the head, hexagonal head, usually passes through the contracted core and enters into the nucleus. The base plate and the tail fibers are held firmly against the bacterial cell, and the hollow core pierces the bacterial. The contractile sheet injects the phage DNA. After penetration, the empty head and the tail of the phage remains outside the bacteria. Next one, synthesis of phage components. This refers to the transcription and translation of a section of phage DNA to make a set of proteins that are needed for the replication of phage DNA. So in this stage, what will happen? Transcription and translation usually takes place. The first products to be synthesized are the early proteins, which are usually enzymes. First of all, enzymes are the ones which are usually synthesized. These early proteins produced by a repair enzyme to repair the hole in the bacterial cell. Second one, a DNA-ase enzyme that degrades the host DNA into precursors of DNA. And third one, a virus-specific DNA polymerase that will copy and replicate phage DNA. Okay, so what happens in the synthesis stage? There is transcription and translation of the phage DNA. The early first products to be synthesized are proteins, mostly enzymes. These early proteins are produced first by a repair enzyme, which is needed to repair the whole bacterial cell. How the whole is produced in the bacterial cell? During penetration, where the bacteriophage is penetrating the alveoli, the whole is formed. Right? So first what will happen? Repair enzymes are first produced. These repair enzymes first close off the hole. Second one, a DNA enzyme. What is this DNA enzyme do? It usually degrades the host DNA into precursors. Third one is DNA polymerase. What is this DNA polymerase do? The degraded host DNA are there, the precursor phase DNA are there. It makes a copy of these phase DNA and it replicates these phage DNA to produce large number of. During this period, the cell energy generating and protein synthesis ability are maintained, but they have been supplemented by virus. The result in this synthesis is that several copies of phage DNA are produced. Okay, so this is what happens during the early synthesis. Proteins are produced mainly. Next, after this, next is the synthesis of the late proteins. The next step is the synthesis of late proteins, which include protein subunits, head and tail of the phage. Each of the several replicated copies of the phage DNA can now be used for transcription and translation of the second set of proteins called as late proteins. Late proteins are mainly structural proteins that make up astromeres and various components of tail cell. Late protein also consists of lysozyme that will be packed into the tail of the phage, which can be used to escape from the host wall during the last step of replication. Okay, understood all of you? So, what is the synthesis? Next one, first one is adsorption, means attachment. Second one is penetration. During penetration, the genetic material, that is DNA, enters into the mixture. Next one is synthesis. What is the synthesis? It consists of transcription, translation, all processes. So, during this synthesis, early proteins are there and late proteins. Early protein consists of types. Late are structure. Early proteins, three types are there. One is required first to close the hole which is formed in the bacterial cell wall, which has formed during penetration. Second enzyme is needed to degrade the DNA and form progeny DNA. The third enzyme, polymerase, is required to form several copies of newly formed DNA material. Okay, so as a result, what will happen? A large number of DNA copies will be. Okay. Next, after this, what will happen? Late protein synthesis. Late protein synthesis are mainly structural techniques, mainly structures which consist of bone, tail, and head, all of this. Okay, not only this, and one of the late protein is also lysozyme, and this lysozyme will be packaged in the tail of the phage, so that when these daughter phases, after they form, this lysozyme will usually help these daughter phages to come out of the bacterial cell wall by causing lysis of bacterial cell. Next one, assembly and maturation. What is this? Having replicated of all their parts, they are called an assembly process. Phage DNA, head protein, and tail protein, all which are synthesized separately in the bacterial cell, all of them assemble together and form a mature infected phage particle. And this process is called as maturation. Last one is release of the progeny phase. Release of the progeny phase in a lytic cycle takes place by lysis of the bacterial cell wall. The bacterial cell wall is weakened and it acquires a spherical shape. This results in the release of mature daughter. The viruses arrange their escape from the host cell during the assembly process only. If while they are in this stage in the assembly process only, they will all arrange themselves how to escape from the host bacterial cell. While the viruses are assembling, lysozyme is being produced at the rate of part of this lysozyme is used to escape from the host cell by lysing the cell wall put together from the this accomplishes the lysis of the host cell and release of the mature viruses to the nearby cell, infect them, and complete the cycle. Okay? And the life cycle of the phage usually takes 25 to 25 minutes to complete because the host cells are ultimately killed by lysis. This type of viral infection is called as lytic infection. Lytic cycle. Okay, so students, have you understood lytic cycle? First one is absorption. Next, penetration. Next, which consists of about transcription and translation. During this stage itself, early proteins and late proteins, all of these happen. Next is assembly and maturation. All of early protein, late proteins, all of these are formed separately. Now, in assembly and maturation stage, what will happen? All of these will come together. One head, one tail, material will come together. Another head, another tail particle will come together. So a large number of pages will form. They will become mature. In the last stage, what is this? Release of the mature phases. Okay. The release of the mature phases is usually helped by enzyme lysozyme, which is usually produced during the late 6th. Okay, this lysozyme usually helps in the lysis of bacterial cell wall so that the daughter cells can come to the cell. The released daughter cells can infect other new bacterial cells. Okay, so this is that is it. Next, temperate or lysogenic cycle. is when growth results in integration of the phage DNA into the host chromosome or stable replication of the plasmid. Most of the gene product of the lysogenic phage remains dormant until it is induced to enter the lactic. Phages enter into symbiotic relationship. So, in this type of cycle, they enter into the symbiotic cycle. They do not kill the host cell. That is why it is not called as temperate or light. I'm sorry, it is not known as light. Okay. Following entry into the host cell, the temperate phase infected becomes integrated with the bacterial chromosome. Okay. Now what will happen once genetic material enters the host cell? This nucleic acid becomes integrated with the bacterial chromosome. And this integrated bacterial chromosome is now known as the prophage. The prophage behaves as a segment around host chromosome. Okay, it usually behaves as a segment of the host. It does not act as a foreign. It acts as a cell. Hence, it behaves intravenously. And this phenomenon is called as lysotomy, and bacterium that carries such prophage is called as lysotomy. The prophage confers new bacterial properties to such bacteria. Such a conversion is called as lysogenic conversion. The lysogenic bacterium is resistant to reinfection by the same or related phages, and this is called as super-infection immunity. Okay, so these are bacteriophages. Examples of virulent factors carried by phage different. Next, phage typing. What is phage typing? The specificity of the phage-bacterium interaction is made use in the identification and typing of bacteria. Phage typing is mainly used for bacterial typing. Example for Salmonella typhus, also for Escherichia coli, and in many. Okay. So, last cycle of a bacteriophage, Rhodopsinian, second, and lysogenous. In the case of lactic cycle, finally leads to the lysis, the host. But in the case of lysogenous cycle, lysis does not take because the prophage, which is usually injected into the bacterial chromosome, usually acts as a segment of the host receptors and behaves synchronously along with it. Because it usually behaves like that, it is called as lysogenic bacterium, and such a process is called as lysogen. Such phages can usually confer new properties. And so when it confers a new property, it is known as a lysogenic conversion. Okay? And these lysogenic bacteria are resistant to reinfection by the same or related phases. If they are resistant, they are known as super infection immunity. Next, bacterium, phage, gene product, and phages. Demographic characteristics. So what is this? First one is Vibrio cholera bacteria. The phage that affects cholera is CTX. It produces cholera toxin, which causes cholera. Lambda phage is the one which infects with sugar. Then it causes sugar-like toxin, to produce simulated cellular. Clostridial phases infect Clostridium botulinum to produce botulinum toxin. Colony phase beta infects Corynebacterium diphtheriae to produce diphtheria toxin and ultimately causes T12 phase attacks Streptococcus pyogenes to release ergogenic toxin, ultimately toxic. Okay, so these are all bacteriophages and lysogenic conditions. Next one, antiviral drugs. What are antiviral drugs? Viruses are difficult to kill because their life inside the cells. Right? Where do viruses live? Viruses are intracellular. Right? So, before they grow, they live, they compulsory need in living cells. So, these viruses are difficult to kill because they live inside the cells. Any drug that kills a virus may also kill the cell. Where is the virus? Is inside the living cell. So the drug which that acts on the specific virus ultimately will kill the living cell also. Antiviral drugs are available for many viruses, and viruses controlled by antiviral drugs are found. Cytomegalovirus, hepatitis, herpes, AIDS, influenza, and respiratory syncytial virus. Viruses have no cell wall and are usually made up of nucleic acid components. Viruses contain enveloped antigen, which is antigen, and they do not have a metabolic mechanism of their own and use host enzymes. Certain viruses multiply in the cytoplasm and others in the nucleus. Most multiplication usually takes place before the diagnosis can be made. Many antiviral drugs are usually purine or pyrimidine drugs. So, do you know what are purine and pyrimidine bases? Yes, we have learned in right? As well as in molecular biology. Many antiviral drugs are pro-drugs. They must be phosphorylated by viral and cellular enzymes in order to become antiviral agents inhibit active replication so that the viral don't resist. Classification of antiviral drugs is a very most important classification. Anti-herpes drugs. Iodine, trifluridine, acetylene, racetylene, transacetylene. Okay, so these are anti-herpes virus drugs. These drugs are used to treat herpes virus infection. Anti-influenza drugs, amantadine, imantadine, anti-hepatitis drug. For hepatitis B and for hepatitis C, the drugs will usually banned. For B, lamivudine, adifovir, tenofovir, hepatitis C, fludarabine, and alpha interferon. Next one, antiretroviral drugs. Antiretroviral drugs consist of component types such as nucleoside reverse transcriptase inhibitors. Next, non-nucleoside reverse transcriptase inhibitors, protease inhibitors, fusion inhibitors, CCR5 receptor inhibitors, and integrase inhibitors. So these are the different types of antiviral drugs. Okay. Now, what are some of the nucleoside drugs? Pseudodyne, albudyne, non-nucleoside drugs, maybe reactive protease, proteinated fusion inhibitors, and fluidine, receptor inhibitors, Myra. So these are different types of protease inhibitors. Yep. So antiviral drugs, please learn about these types. What are the different types of anti-herpes virus drugs, anti-influenza virus drugs, anti-hepatitis virus drugs, and retroviral drugs. So first one, anti-herpes virus drugs, most important is acyclovir. How does it act? The virus is usually specific for thymidine kinase, which usually kinases and inhibits herpes virus DNA. As a result, what will happen? It inhibits DNA polymerase. Once it inhibits DNA polymerase, there is stopping of lengthening of DNA strand, and the terminated DNA inhibits DNA polymerase irreversibly. Uses of acyclovir, it is mainly used in the treatment of genital herpes simplex, primary disease as well as recurrent disease, mucocutaneous herpes simplex infection, herpes simplex, keratitis, herpes zoster, and chickenpox. So, acyclovir is used in the treatment of herpes simplex infections as well as chickenpox and shingles. Next, valacyclovir and famciclovir. Both are ester prodrug of acyclovirs. Anti-influenza drug consists of about amantadine and rimantadine. Okay, now what is this amantadine? How does it act? It inhibits replication of influenza A virus, but not influenza B virus. So amantadine is usually specific to stop the inhibition of influenza A, but it does not combat influenza B. It is mainly used as a prophylactic measure during an epidemic of disease. Rimantadine. It is the methyl derivative of amantadine. Okay, it is the derivative of amantadine, and side effects are usually lower when compared to that of amantadine. Next, anti-hepatitis drugs. Hepatitis viruses are many. A, B, C, D, E. Okay, there are so many types of hepatitis viruses. And for those, we can use lamivudine, ribavirin, and interferons. These three are mainly used for the treatment of hepatitis viral infection. Now, what is this lamivudine? It is an inhibitor for both hepatitis B virus as well as human immunodeficiency virus reverse transcriptase. Lamivudine is used as a first-line drug for chronic hepatitis B. Ribavirin, it is a broad-spectrum antiviral agent. It is also used for the treatment of influenza A/B respiratory syncytial virus. Interferon. Interferon is a cytokine, right? And interferon is mainly used in the treatment of hepatitis C, B, as well as AIDS-related Kaposi sarcoma, condyloma, acuminata, herpes simplex, herpes zoster. What is herpes zoster? What does it produce? Shingles, right? So it can be used for the treatment of shingles also, and cytomegalovirus. Next, antiretroviral drugs. Steroidine, stavudine, lamivudine, and tenofovir. So these are all which are used as antiretroviral drugs. And for antiretroviral drugs, there is first line treatment as well as when second line treatment is also there. This is first line antiretroviral therapy, second line antiretroviral therapy. Okay, so that's all for today. For today's class, we have learned about bacteriophage, learn about morphology, the diagram of bacteriophage, and different morphological parts. And next one is life cycle of a bacteriophage, life cycle and lysogenic cycle with diagram. Next, antiviral drugs definition. What are the antiviral drugs and how many types of antiviral drugs are there? Give examples for each antiviral drugs. Example, anti-herpes virus drugs, anti-influenza virus drugs, anti-hepatitis virus drugs, and anti-retroviral drugs. Okay, that's all for today's class. Thank you once again for coming to this class.